I hope you all had a pleasant journey here today. Let me introduce myself. My name is Clydiewel Arsugitan. Today, I will report about cell division, the interphase, and mitosis. Interphase is the portion of the cell cycle that not accompanied by gross changes under the microscope and includes the first gap phase, synthesis phase, and second gap phase. The first gap phase, the period of growth and normal metabolic rules, and synthesis, DNA replicates, and second gap phase, enzymes are produced to prepare the cell division. Mitosis is a fundamental process for life. During mitosis, a cell duplicates all of its contents, including its chromosome, and is split to form two identical daughter cells. Mitosis is divided into 5 phases: interphase, prophase, metaphase, anaphase, telophase. DNA is copied, chromosomes pair up, chromosomes line up at equator mitotic spindle, sister chromatids pull apart, cell pinch in the middle, two identical daughter cells.
Hello guys, this is Maritas A. Gutierrez and I'll be reporting today about mutation. So I have here a mutation is a change that occurs in our DNA sequence, either due to mistakes when the DNA is copied or as the result of environmental factors such as UV light and cigarette smoke. So chromosome mutation is an alteration in the structure and number of chromosomes. So I have here the types of mutation. The inversion, it is the reversing linear order of genes. The deletion is the type of mutation involving the loss of genetic material. And the third is duplication, a type of mutation that involves the production of one or more copies of a gene or region of a chromosome. And the fourth, it's translocation, an exchange selection between two or more chromosomes. Okay, so I have here the types of change in DNA. So class of mutation, it's a chromosomal mutation. And a type of mutation, of course, it's inversion. And the disease that may occur or may link using this mutation is the Optis-Caveglia syndrome. And the deletion is Sindu-Chart syndrome. Duplication, it's link for some cancers, and the translocation is one form of leukemia. So I've here the examples or the examples of a mutation, chromosome mutation. Okay, so deletion is a chromosome segment lost. Translocation is a segment from chromosomes is transferred to another. And the duplication is a segment from one chromosome is transferred to its homologous chromosomes, given it is duplicate of some genes. And this inversion is a segment of a chromosome and it's inverted.
Hello everyone, my name is Lovely Domingo. So ang topic po natin, change affecting number. Haploid D. One set of parental chromosome is odd cells. A cells that contains a single set of chromosomes, the term haploid can also refers to the numbers of chromosomes in eggs or sperm cells, which are also called gametes. Gametes contain half chromosome contained in normal diploid cells of the body, which are also known as somic cells. So diploidy, two copies of each chromosomes, and polyploidy, more than two complete sets of chromosomes. Polyploidy, one set of parental chromosomes are double or triple. The condition in which a normal diploid cells organism requires one or more additional sets of chromosomes. In other words, the polyploid cells organism has 3 or more times the haploid chromosomes number. Types of polyploids: autoploids and alloploids. Monosomic, loss of individuals' chromosomes, is the state of having a single copies of chromosome where instead of the usual 2 copies found in diploid cells. trisomy presented with additional chromosomes. The presence of an extra chromosome sum of all body cells, this result in a total of 3 copies of the chromosomes instead of the normal 2 copies. Non-junction. Due to the failure of homologous chromosome to separate causing one or more extra chromosomes. Failure of 5 chromosomes to separate during cells division, so that both chromosomes go to one daughter cells and none go to others. Examples of non-disjunction: Down syndrome, triple X syndrome, Klinefelter syndrome, and Turner syndrome.
I'm Princess Diane Pilien. My report for today is about metaphase. So now, what is metaphase? Metaphase chromosome is at the equatorial plane and connected to the mitotic spindle. What happened during metaphase? During metaphase, the kinetochore microtubules pull the sister chromatids back and forth until they align along the equator of the cell called the equatorial plane. There is an important checkpoint in the middle of mitosis called the metaphase checkpoint, during which the cell ensures that it's ready to divide.
The next stage of the mitosis is the metaphase. All chromosomes are located on the equatorial plane, but 2 filaments of the spindle are attached to their centromeres. At this stage, the chromosomes have reached maximum thickness. It's also the longest stage of the mitosis. Next is the anaphase. Anaphase. It's aligned double chromosomes that begin to separate from each other and move to different poles due to pulling filaments. Genetic material in the chromosome switch was doubled during the synthesis phase of interphase. It's now halved so that each daughter cell has the same complement of DNA as did the G1 cell. It's also a fast stage of the mitosis.
Hi, my name is Salve. So today I will be discussing all about the telophase. So we all know that telophase is the final stage of mitosis. Its name was derived from the Latin word telos, which means end. So telophase is actually the reforming of the nuclear envelope around the separated sister chromatids and the cleavage of the cell. So to begin the process of telophase, it actually begins with the replicated paired chromosomes that will be separated and pulled to the opposite side. So this is what happens. It will be pulled towards the opposite sides, and then it will be followed by the nuclear membrane that will be formed in each set of chromosomes to separate the DNA from the cytoplasm. So this is the nuclear membranes, and then it will be followed by the chromosomes will begin to uncoil. This will make them diffuse and less compact. And then along with the telophase, the cell also undergoes a process called cytokinesis that divides the cytoplasm of the parental cell into 2 daughter cells.